Hello and welcome to this video on how animals act as a source of disease even when we think the disease has been eradicated. There are many viruses, fungi, bacteria, prion proteins and more that at one point or another humans had thought they removed permanently. Often we have been right, but there have also been exceptions. Two examples of this include tuberculosis and plague. Tuberculosis was gradually removed over time, but in some countries people are still infected. It turns out that badgers can act as a reservoir for this, and when they come in contact with humans and cattle, they can then spread tuberculosis back into the population. A much older example includes plague. Most people have some knowledge of how plague spread in the medieval age in Europe. Rodents brought fleas from Eastern Europe and the Asian steppes. These fleas carried plague to humans from the rodents. When the people died and the disease was unable to spread, the rodents went wild. When the humans recovered to adequate numbers, the rodents came back and shed the fleas again. This cycle repeated many times. While these two examples show how a disease can keep recurring, it's not just existing diseases. Animals can and often are a reservoir for novel diseases. A reservoir like this is called a zoonotic reservoir. SARS, COVID, MERS, anthrax, Ebola, various parasites, and more were all novel diseases. That is, a disease that humans have not encountered before and therefore were unprepared to handle. There is a rough estimate that 6 out of every known current infectious diseases that we have to deal with can be spread from animals, and 3 out of every 4 new disease will come from animals. That means the vast majority of what we're currently trying to compete with, in a pathological sense, is going to be caused or come from animals. The general process that these diseases follows is a circle of events. It begins with the reservoir. That is going to be the animal in question. Badgers, rats, bats, or whatever other animal it is. The pathogen generally does not kill the host, but it can make them ill. Generally, even in that case, the disadvantages to the animal are not so great that they die out. The animal will then come into some kind of vector for infection. This can include water or soil, or it could be direct physical contact or even bodily fluids. Now that the pathogen is out of the reservoir, it can find a way to enter a human host. If it finds a susceptible human host, an infection occurs. At some point, the disease is released back into the environment where it finds its way back to the water, soil, or food the animals feed on, and infection can occur again. This creates the circle you see here, and it is effectively the cycle of life, but for diseases. This cycle, or another animal with the same disease encounters a human, and the cycle occurs again. While it is one way you can look at this, it also occurs that you can acquire diseases from things like food. For example, parasites like tapeworms in undercooked pork can be transmitted from the animal to the human by the food itself. It can come from water contaminated with feces, and this then means that the bacterium, virus, or parasite gets into the human GI tract. There are any number of ways the pathogen in question can have a vector that's not as obvious as simply being exposed. Things like fleas and mosquitoes are quite common, especially mosquitoes because they are so effective at spreading diseases. Direct contact is generally far more obvious. For example, touching a diseased animal, you can then acquire the disease. This is how we got vaccines using cowpox. It is transmitted in this way, and it was then realized that it imparted a degree of immunity. Indirect is more difficult. This is often done by touching the habitat of an animal, like for example fish tanks and the water therein, or other housing for domesticated animals or wild animals that have made their home in the domesticated environment. The last of these is especially common, or at least increasingly common, as humans move more into areas where wild animals are present, or semi-domesticated animals like rodents increase in numbers as the density of humans rises. The more people there are living in one area, the more opportunity they have to get food from them in one form or another. This is why New York City is notorious for its rats. The problem with that is that it increases the number of possible exposures. This therefore increases the probability of a pathogen jumping between species. This step is key. A zoonotic disease needs to be able to jump from an animal to a human, otherwise the animal is not so much a source of the disease as it is simply a diseased animal. This often occurs in combination with how diseases appear and develop in animal reservoirs in the first place. Since most diseases that we're aware of now seem to come from animals, and because they're infecting humans, we need to have some sort of mechanism that bridges it, we know that there's certain common traits. Mammals tend to be a more serious issue as they share more similarities to humans. Therefore, the chances of something infecting them infecting a human are much higher. This then also becomes true, for example, if we're looking at carnivores and non-human primates. There's even good evidence showing that rodents and ungulates can also have this effect. All of them generally have some sort of similarity to humans in some way. This could be a receptor on the cells, a gene that's used for a particular purpose, a biological process, diet, and many other mechanisms. The animals are already using this as part of their biology. It's part of their life. Now, a small mutation can occur, and suddenly, not only can it use the existing version of whatever it is in that animal, but it can also make use of the human equivalent. This is what allows the disease to jump between species. This can occur especially if you are in a species that has a very unstable genome. Bats are notorious for this, and it's why viruses can often mutate within them. Another option that relies less on random chance is the recombination of genomes from multiple pathogens. This occurs when a host animal is infected with two different diseases at the same time. The two share a gene or mechanism, and this is often a viral event. The most common example being when a virus enters a cell, a second virus enters the same cell, and now the genome of each gets mixed together during replication. That leaves the virus with bits of DNA from each of the particular pathogenic viruses that have entered. These bits can include genes that provide an advantage. You go from having two completely distinct diseases to having one disease that has traits from the other. This can be things like being more infectious, being able to take advantage of a receptor, producing particular markers that help it to avoid the immune system, and more. The final consideration, especially zoonotic diseases, is migratory animals. Animals that move large distances often carry the issue that they will spread a disease over a very wide area. This increases the exposure in those terms, and it means there are multiple species that could be exposed to it and become vectors in turn themselves. Migratory animals and birds are the most high risk of these, and things like avian influenza are a worry because of it. Avian influenza may be present in one location, but it can then be spread over a huge area, and then affect domesticated poultry, and from there humans. The movement between one animal reservoir to the next and then humans is often facilitated by that genetic recombination, and this is why animal-specific viruses can become human-specific viruses. At the same time, not all diseases are one way. For example, zoonotic reservoirs can be either uni or bidirectional. That is, either the animals can infect humans, and that's how it ends, or the animal can infect the human, and the human can in turn infect the animal again. This last area is a big concern. So long as you have that constant to and fro with humans to animals, not only do you have the risk of keeping the animals infected, thereby continuing to act as a reservoir, a wild source for diseases, you also have much more difficulty in getting rid of it permanently. It's very hard to stop the complete spread from humans to animals, whereas animals to humans is a little easier to break, by simply following good, basic, standard hygiene. This is a very simple summary of how animal reservoirs of disease work. They are simply animals living their life and carrying something that finds a way to jump from their species to ours. And at that time, it goes from being an unfortunate situation for that animal to being a really big problem for us. And it's why we have so much trouble in dealing with them. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions you have below.